So this particular drug, TPI 287, is a member of the taxane family. It was actually derived from understanding changes that are needed in taxanes to make them work better. Research over the last 10 to 15 years has really been focusing on new drug development in making drugs that will work better, smarter, compared to what we've learned from tumors and how they are able to bypass some of the particular drugs that we're using today. So this one, which is a member of the taxane family, which are microtubulin binding proteins. These are different than most of the other microtubulin drugs we use today, which are actually inhibitors of it. This actually binds and stabilizes microtubulin. Modifications were made to native taxanes to change both the side chain of the drug as well as the bacitracin ring structure. Changes in the side chain make the drug more lipophilic, which allow the drug to get in and stay in cells longer, and the changes to the bacitracin ring help them circumvent the multi-drug resistance protein 1 transporter system that are known to affect a lot of cancer cells, as well as allows it to bind to mutant tubulins, which can be formed by a number of these cells. So the bacitracin ring itself is this portion. The first taxane developed was paclitaxel from the western yew tree more than 40 years ago. This structure, you can see, was changed in a number of places to make this new compound for TPI 287, and then the side chain structure was changed as well, which has allowed it to be more effective. So why was that helpful? The taxanes in general are not helpful, are not effective in neuroblastomas. Though they've been around for almost 20 years in clinical practice and are helpful in a number of adult cancers, they really haven't been very effective in neuroblastoma cell lines. So this particular drug, after it was developed, was tested in multiple cell lines, and one of the ones that was found to be fairly effective in was in neuroblastoma. So with that, you can see on the left side a relatively sensitive neuroblastoma cell line with increasing concentrations of TPI. Also, along with temozolomide, there is some synergy. It's a little difficult to tell when it's a very sensitive cell line, but in a very resistant cell line, you also see effect of both TPI and then a synergistic effect with temozolomide. The TPI was then looked at in a number of other cell lines. One of the biggest problems for the taxanes, as I said, is this problem with multi-drug resistance. So in both a multi-drug resistant negative cell line and a multi-drug resistant positive cell line, the effects of TPI were compared to standard dosings of vincristine, and found that in the multi-drug resistant negative cell line, there was a significant increase benefit, but in the multi-drug resistant positive cell line, there was actually a tenfold increase in effectiveness. There was not a significant difference, though, between the two different cell lines, so the effect was fairly equal across different types of neuroblastoma cell lines. From this, then, it was compared in animal models to see whether or not against other taxanes you still saw the same effect, and here tumor volume size and amount of time to progression were assessed, and the different taxanes versus controls, and you can see that TPI had the best effect in keeping the cells from growing, the tumor cells within the animals from growing for the longest periods of time. So from this, Archer then approached Giselle and said, well, I think this is probably a pretty good drug. We probably ought to do something with this in neuroblastoma. So we brought it to our consortium. As I said before, the taxanes are historically poor activity against neuroblastoma, so they really haven't had much use in clinical practice. So TPI was then selected on this basis of high potency in preclinical studies to start a phase 1 study. So the phase 1 study is of TPI 287 as a single agent drug, and then in combination with temozolomide, because there was shown activity in the preclinical studies for recurrent and refractory neuroblastoma and medulloblastoma. And this was the NMTRC 001 study. This study had primary objectives to understand the safety, tolerability, and then find the maximum tolerated dose of the drug. This is the benchmark for a phase 1 study. It's important to understand that in a phase 1 study, the most important aspect of the treatment has to be finding out how safe the drug is and whether it's effective. So you need to find out what side effects there are, what actually may go wrong, and any clinical benefit from the drug has to be a secondary objective. So then the secondary objectives here were to find out as a single agent and then in combination with temozolomide, any overall response, progression-free survival, as well as median time to survival. In addition, we were very interested in understanding the pharmacokinetics, or how much of the drug is hanging around, how long it's hanging around, to understand how well it's working within the patients. And then a secondary objective, which has been for all of our studies, is to be able to collect biologic samples from patients, either from tumors or bone marrow, to test those in the lab, both for the particular studies we're working on and future studies, to have material around to really start understanding better what happens for these tumor cells compared to pre-made cell lines, which are very different than a lot of the tumors that we actually see in patients. So the study was an open-label, multi-center dose escalation study. Dr. Roberts did a very good job of explaining what a dose escalation study is. In recurrent medulloblastoma and neuroblastoma patients, the first 2 cycles of the therapy, which are 28 days long, were drug alone, so that we could understand what this drug was doing, be able to get safety data on this drug, as well as get pharmacokinetic data for this particular drug. The drug is given on weekly doses as an IV infusion on days 1, 8, 15 of a 28-day cycle, so there's 1 week off out of a 28-day cycle. The starting dose of 90 milligrams per meter squared was chosen because it was 2 dose levels below what the maximum tolerated dose was that was found in adult trials that have already occurred. The cycles then 3 through 6, and actually subsequent to that if patients were doing well, combine TPI 287 with temozolomide. Each of the cycles were given the same. You had TPI 287 given on days 1, 8, and 15, and temozolomide itself was given days 1 through 5 of a 28-day cycle, which is a fairly standard dosing. Pharmacokinetics were still collected also at the beginning of cycle 3, so that we could understand the interactions potentially between TPI and temozolomide.